Hello guys, myself Dr. Atul Nagpuri and I am at CSIR-National Chemical Laboratory, Pune in Dr. Gopinath's laboratory. And I am happy to inform you that we have with us Dr. Devaraji, Senior Research Associate to CSIR-NCL Pune, and he is going to explain us how surface profilometer works. Please sir.
Yeah, many thanks for Dr. Atul Nagpuri for introducing me. Myself, Dr. P. Devaraji at National Chemical Laboratory, CSIR-NCL, under supervision of Dr. C.S. Gopinath. And I did Ph.D. at CSIR-National Chemical Laboratory. Then after I went for my Post-Doctoral research, South Korea. Then after I went to China. Now again, I started here Senior Project Associate. So now I am going to explain the importance of profilometer, surface profilometer. Actually here we are doing the photocatalysis. So in the photocatalysis, so we are using powder catalyst as well as thin film under direct sunlight. So we are doing the reaction. So before the reaction, the importance of this our work is that the amount of the catalyst that should be optimum. So here we are having the profilometer, surface profilometer. So the thickness of the samples coated on the FTO or some glass material that we can analyze. So here you can see that you can maximize the things actually. So this is the sample. So this is the sample FTO plate. So over that we are coated the titanium dioxide or some other material. So you can coat actually. So the thickness between this slide as well as the sample you can analyze from the surface profilometer. So yeah, this is the holder. You can keep the sample here. It's a very sensitive instrument, so we have to be very careful. And so this is the window. So here this is the software. From here I am starting the measurement actually. So you can see the profile, the surface, the roughness and the smoothness of the samples you can analyze. So yeah, this is the cursor. So it is dragging on the sample, and this is the FTO plate. So then it is running this surface analysis. So it's moving on. Right. So as this ended, so we have to optimize the position of the sample. So here you can again. So here there are the adjustment is there and the key is there. So from that to and fro, you can adjust the this position of the samples right now. So now we adjust that. Again we are starting the sample. So it's measuring the surface and the smoothness as well as it's analyzing the sample. So you can see on the window directly from the monitor actually. So this technique is very important, and so we know the thickness of the samples. That is a very useful for the our reaction actually. So now the analysis is done. So here nicely the surface profile has come actually, profile of the sample. So here reference is there as well as our the material surface, the surface profile is there. So here we can analyze that the surface thickness of the sample is 7.07 micrometer. So these the data which we got from this instrument. So this is a very sensitive as well as this the thickness of the sample is around 7 micrometer, and 8 micrometer of the sample is very suitable for the photocatalysis. So the light has to penetrate, as well as after that the photon, electron and holes are generated to the surface. Then the reaction is happen on the surface. So the thickness should be optimum to the 8 micrometer. Now it is 7 micrometer. So this will be close to the 8 micrometer. So with this sample we can go ahead for the other reactions.
So see guys, Dr. Devaraji has nicely demonstrated how the surface profilometer can be worked and can be extensively used for surface science technique. Thanks for watching. Thank you very much.
Yeah, thank you for coming to
Devaraji.
Thank you.
Guys.